Hello, my name's Rick Hall and I'm going to do a presentation from the bioinformatics field, a comparison of two pipelines, Mothur and Dada2, which is from my placement at Menzies School of Health Research. So why pipelines? Well, in the field of metagenomics, the application we use tools to study the genetic material from environmental uncultured microorganisms through next generation sequencing. And in this case we use 16S RNA, which allows us to see the bacterial diversity and detect thousands of microorganisms. The process of sequencing produces artifacts that are unrepresentative of samples, and these have to be removed reliably to provide clean data for studies. And this uses well-constructed algorithms or pipelines that untangle the sequences into useful and reliable data. This study compares two pipelines, Mothur and Dada2, using upper respiratory samples, the sequences that were generated through NGS, and it's a small, well-studied subset of a bigger set. Of the two pipelines, we're going to have a look at Mothur first. That's the oldest program. It groups sequences into OTUs, and it's an open source platform supported by a lot of users. Dada2 is an improved version of a 2012 release. Dada2 was released in 2017. It's also open sourced. It's based on R code, and it uses amplicon sequence variants rather than operational taxonomic units. So they're both open source. They both are well supported by user groups, and both of them sort and filter the sequences. Mothur uses OTUs. Dada uses ASVs. Mothur is only for 16S RNA, whereas Dada2 can be used for just about any gene sets. Mothur denoises sequences via arbitrary user-defined parameters, whereas Dada2 uses error algorithms to denoise the sequences. Our data consists of 20 samples, 12 of which are from paired runs of 6 samples. Some are constructed, and all are quite well studied. 8 samples were no template negative controls. The data size is about 1 gigabyte from the full set, which is of 20 gigabytes. Let's have a look at Mothur first. I ran that using Cheetah, which is the high performance supercomputer that's a shared resource between Menzies and Charles Darwin. It has more than 400 processing units and a pretty powerful 3 terabyte of RAM. And it uses a Linux-based command line language system. The main requirements when working with Cheetah are a portable batch system, a group of commands that that allocate the job and show it where to email the results to or its status, a working directory from which data is taken from and output to, and the program, the group of algorithms. Within Mothur, the pipeline, it starts off by identifying the directory. It screens the sequences, so from predefined parameters such as homologs and the number of base pairs. It aligns the forward sequences with the reverse ones, then it screens them again if they're outside, if they're too long or too short, or again, different things. Using R code, then it reformats these files for the second part. Then it refines and removes the chimeras. It pre-clusters and assigns taxonomy, filters again, clusters with 3%. We ran it down to 2% as well, similarity, and creates files for the final outputs. This is the coding script. That's all of the whole program that was used for Mothur in Unix, apart from this small part here, which was the R code. This is the portable batch system, the group of commands that tells Cheetah how to start off with this program. In blue are the notes, that they're not run as the program. Cheetah recognises that they're not part of the program, and in black here we've got the actual line commands that run the program, the algorithm. It runs through a GUI, a graphic user interface. This one is WinSCP. And in this program you can put the files. These are the FASTQ files that were used in this study. And the command line instructions are run through this little sub-program called PuTTY. So Dada2 ran in RStudio on a laptop to start with, and again these blue are notes, the other are the program. This screenshot shows the first output of Dada2 down here, and this is the highlighted section was the code that was run to produce this map, those plots, I beg your pardon. This is the console where the actual lines that are executed in R appear. Over here we've got the environment panel. You can do other things in here as well, and these let you know what values and variables have been produced as the program runs. And here are the first plots generated by this line of code. I might mention that they're not necessarily viewable. They weren't viewable in Cheetah like this. These two plots show a characteristic of sequencing. These are the forward reads, and you can see they're pretty good quality until they start to drop off a little bit later on. And if you compare these to the reverse reads, that's another characteristic again of sequencing. They're much poorer quality. The quality score isn't as high, and they drop off quicker than before. This is the guts of the Dada2 program, the error algorithm. It plots the data. It constructs functions to match that best match the data, and through that it helps find the outliers, the noise of the sequences that aren't necessarily going to be good data. And when Dada2 is complete, these are all the variables that have been produced, all the values. But when you close RStudio, you lose all this. So it's important to find a way to keep that data. So I've added in 4 more lines of code, and they produce files in this part of the window, and they're available through your File Explorer or your normal computer program. You can find these files, and these are constructed in a way that will translate to Excel files and such quite easily. Dada2 has a feature where that information is quite easily slots into another package called PhyloSeq, which generates useful information. This is a family taxonomic bar plot for the samples down here, showing you the various composition of those samples. And here's a heat map again. You've got the samples down here. It says OTUs, in this case it's the ASVs, and you can see where the similarities lie in that lot of samples we ran. I later ran Dada2 through Cheetah as well, but I had to run it through the head node, and you have to be careful when that happens because the head node is the only one that has the internet access, but it can bottleneck everything. These are in brown when I was running the interactive mode for the Dada2, and I had to keep an eye on this to make sure I didn't overload the system and to upset the other users. Here I ran it closer to Christmas, and when there was no other traffic, and I ran the 20 gigabyte full set, and you can see here how, and we fixed up some stuff with Cheetah to run R. You can see how here it's shared the load through several nodes, and it's multi-threading, multi-processing that data, so it gets through a lot of data quite quickly. Let's compare that pipeline outputs. I've done it 3 ways. The first one is accuracy qualitative. The first takeaway is the number of OTUs that are found. Dada2 is quite clean. You can see it's found less of them, and it has less unexpected. They both found all the 20 that were, or varieties that species that we expected in that sample, and the relative abundance of the first unexpected was only just slightly higher with Dada2 than it was with Mothur, but Mothur had a lot more unexpected. Here we've got the same table and another table for the same sample where we've got the total number of reads, and they're quite similar between Mothur and Dada2. Dada2 has less unexpected than Mothur. It's a much cleaner data set that's produced. The second of the 3 samples that we're comparing here is 783. This varied in relative abundance. It had 20 species, but they started at around 22% relative abundance and went down to 0.02 relative abundance. Each of the programs found 16 out of the 20, So they only found one of the 0.02, and that was the species that overproduces through the PCR. And that was at 0.04%. But again, Dada2 is much cleaner. Here you see the unexpected reads compared to Mothur Dada2 is much cleaner data, and they both found 16 out of the 20 samples. They both found the same. And the third sample that we're comparing was quite a variable mix. Again, with Dada2, hardly any, no or zero unexpected reads compared to quite a few within Mothur. So the take home from there is that Dada2 has cleaned the data better, and both lines accounted for most of the expected taxa. The second way was looking at the relative abundance, how well they reproduced. Here we've got 782, where the samples were at 5%. They've found, both of them have found pretty much this very similar quantities. Staphylococcus wasn't very well differentiated between either pipeline. There were two species there amounting to 10%, but neither pipeline distinguished them very well. Bacteroides is overrepresented, which is a feature of the way it behaves under PCR, and Pseudomonas was underrepresented, which again is a feature of the way that it behaves under PCR. Here we've got the second sample, where we start around 22% and we drop off to 0.02% of expected relative abundance. And you can see that of the 16 samples, both pipelines have got quite similar results there. And again, with the last sample, we've got quite a variable mix, but both programs. What is important here is that Streptococcus wasn't very well differentiated within Mothur, but it was very well differentiated in Dada2. We'll get to that. That's an important feature. So the take-home is that neither pipeline distinguished Staphylococcus. They overrepresented Bacteroides and underrepresented Pseudomonas, which is a feature of the PCR, not the programs. And most of the other relative abundances were similar reported. Of the last of the three, reproducibility, I've done this two ways. One was with tables. These are quite similar. I'll go to another way quickly so we can see that data better. This is using denograms. This is the denogram for Mothur. Similarity down here would be 100%, so that these are paired samples, and up here 0%, so these are not similar to those. We'll get them both up together so we can have a better look. There's 6 paired samples, which 5 of which are very similar between both programs. There's not much to see between them. They've both got these paired samples quite close, except for the 6th one, which isn't much between them, but you can see Dada's got them more similar than Mothur has, and the rest of them were negatives, and they're quite random, as you'd expect. So to conclude, Dada does appear to be a great alternative to Mothur for producing the taxonomic tables from the 16 sRNA that we used. Dada2 denoises the samples better, and it distinguishes Streptococci species better, which is significant. Dada2 can separate reads with less than 2% variation and still control for errors. And in samples where distinguishing these fine variations is important, such as single nucleotide polymorphisms, etc., Dada2 with its amplicon sequence variances and set of operational taxonomic units enables distinctions that Mothur can't. It's a great alternative. I'd like to thank Dr. Robin Marsh. She was awesome. Really enjoyed this placement. And thank you to Dr. Natalie Millick for matching my character with this placement. It was a real pleasure, thank you. And thank you also to my media aide, Jack Marsh. Thank you and bye.